Biomedical wastes are wastes that are generated in a healthcare facility during the process of diagnosis, treatment, or immunization of human beings or animals, or during research, or during production of any biological materials. The latest biomedical waste management guidelines that we are following right now is of the 2016 rules, and the latest amended rule is 2019. According to the 2016 rules, the 10 categories of biomedical waste have been compressed and segregated only into 4 categories, that is yellow, red, blue and white. In short, waste that can be thrown or segregated into a yellow color-coded bin are basically waste that are contaminated with blood or any body fluid, including blood bags. In the red category, we separate only the biomedical wastes that are plastic in nature and are recyclable. In white, we include metallic sharps. In blue, we include glass, broken glass and metallic implants. The basic steps of biomedical waste management are segregation, collection, pretreatment, transportation, storage and disposal. Segregation is a step that is being done at the time of generation or at the source of generating the biomedical waste. The step of segregation is separating the different types of biomedical waste according to its category, that is yellow, red, blue and white. After segregation of the biomedical waste at the point source of generation, waste can be collected at least once a day, or when the bins are full, up to 2/3. During collection of biomedical waste, the waste bags should be tied firmly with the help of a zip tie or any kind of thread or rope available. It is advisable not to use a staple or a stapler, since it may cause injury to the person who is collecting. It is advisable that during collection of waste, use double bags for the safety of the designated person who is carrying the waste. All categories waste should be collected, tied, double bagged at the same time and carried from the point of source generation to the point of pre-treatment using a covered trolley if available. The time of collection should be during the time of day where there are very less traffic, usually early in the morning or late in the evening, where there are less patients or staff as much as possible. When reaching the pre-treatment area, each color-coded bag should be weighed in and noted in a respective biomedical waste logbook. In the logbook, you will have each column dedicated for each category bags. For example, yellow, blue, red and white. In each column, for example, under yellow, you will have the number of bags generated from the facility per day, the total weight of the yellow bags generated per day, and the name and signature of the designated person who collected and transported the biomedical waste to the pre-treatment area. For red bag, we either use the chemical method of pre-treatment or we use the autoclaving method for pre-treatment. If the facility uses a chemical method of pre-treatment, then freshly prepared 1% hypochlorite solution is to be used. If autoclaving method is to be used, then the red waste is to be autoclaved at an appropriate temperature and pressure for at least 1 hour. For facilities opting for autoclaving method, the temperature to be used is at 121 degrees Celsius at a pressure of 15 psi for at least 1 hour. After autoclaving, the red bag is then shredded with the help of a shredder, collected in a new red color bag, zip tied and labeled. Pre-treatment for yellow category waste that comes from the microbiology section, especially medias or cultures, we only autoclave it. Rest of the waste generated, for example, like tissue, gauze, cotton or blood bag that have been contaminated with blood, can also be autoclaved or disinfected using non-chlorinated compounds such as aldehydes, phenols, etc. Chemical waste that is generated either from the lab or from the drug section, like expired drugs or expired cytotoxic drugs, are also discarded in a yellow category bag. Such waste do not require pretreatment. They are to be discarded in a yellow bag, tied with the help of a zip tie, labeled with a biohazard symbol, along with a cytotoxic waste symbol, along with the chemical name outside of the bag. For blue waste, intact glasswares can either be sterilized or disinfected using 1% freshly prepared hypochlorite solution, and thereafter can be washed using a detergent and distilled water. For broken glasswares, we sterilize it or disinfect it using 1% hypochlorite solution, and then we dispose it in sharp pit. For white category waste, metallic sharps fall under the white category. Example, needles discarded from a used disposable syringe. We can sterilize it or disinfect it in 1% hypochlorite solution and then dispose it in a sharp pit. After pretreatment of the yellow waste and the red waste, we transport the yellow waste and red waste in double bags, securely tied with a zip tie and labeled, using a covered trolley into a temporary storage area. This temporary storage area should be secluded, well confined and designated area away from human traffic and the lab, the OPD areas, IPD areas, etc. The temporary storage area will be under lock and key, and the key will remain only with one designated person. Waste in the storage area will be collected for not more than 48 hours. Collection of biomedical waste from the common biomedical waste treatment facility or the municipal board will be done within 48 hours of collection of the biomedical waste. Information should be given to such entities regarding the collection of the biomedical waste generated as soon as possible by a designated biomedical waste nodal officer. A logbook of biomedical waste management should be maintained in each department, in each section, in each room, wherever a biomedical waste is generated. And a common biomedical waste logbook should also be maintained and handled by the designated person who's holding the key to the common storage area. Lastly, for waste of the blue category and white category, the final disposal is in the sharp pits. An important note, sharp pits should also be well maintained, well fenced, protected from stray animals. It should be supervised and seen that only sharps such as metallic sharps or glass sharps are thrown into the sharp pits.